So hello everyone. So I'm Hairong from Center for Environmental Biotechnology, Bangor University. So our lab for this project is focusing on hydrolytic, discovery and engineering hydrolytic bacterial defluorinases. So we are aiming to expand the substrate range rather than only fluoroacetate, but for more degradation on polyfluorinated compounds. So, as we all know, we have several groups of enzymes. They have reported or proposed defluorination activities. For example, the fluoroacetate dehalogenases, haloacid dehalogenases, and these monooxygenases. So our lab is focused only on the hydrolytic ones. So like the monooxygenases, they replace the fluoride ion with this hydroxyl group, but they don't need the cofactors and the electron donors for the hydrolytic one. And we also have the proposed or the potential fluorination enzymes like reductive dehalogenases. So this is a large oxygen-sensitive enzymes. They have a iron sulfur cluster, and this is really hard to express in the recombinant E. coli, and it's hard to purify for the biochemical characterization. And also we have a peroxidases. This is a heme-containing proteins. They use a hydrogen peroxidase for the reaction. And also we have lacases. They contains 4 copper ions as a cofactor and use a molecular oxygen for the reaction. So that's all the proposed enzyme we are having for the defluorination reaction. So for the our lab's work, they initially conducted in the University of Toronto and later happened in the Bangor. So our focus is to discovering the defluorinating enzymes from sequenced bacterial genome. So for doing this, we are doing a silicon mining and activity-based screening. So we are looking for the enzymes they are defluorinating and from the alpha-beta hydrolase superfamilies. So we identified 9 bacterial enzymes with the defluorination activities against the fluoroacetate. So the fluoroacetate is the most abundant organofluoride compound in the nature, and they produced by the streptomycin as a secondary metabolite, and they also produced by some of the plants as their self-defense mechanism. So back to the paper. We have 9 discovered, and there are 5 from fluoroacetate dehalogenases and 4 from the haloacid dehalogenases. And this paper was published in 2010 and in the microbial biotechnology. So later, we focused on the biochemical characterization of fluoroacid dehalogenases. So they form Rhodopseudomonas postris, so which is a photosynthetic bacteria and well known for its metabolic versatility and potential in bioremediation. So for this enzyme RP1163, we are lucky to get this crystallized, and we also got a series of high resolution crystal structures during this defluorination reaction. So we have the first one, the overall structure. So this is the alpha beta hydrolase 4, and then we have a mycalase complex, and we have this covalent intermediates, and we have this enzyme product complex here. So also for this study, we discovered this. This RP1163 not only have activity on the fluoroacetate, but they do also have activity on the bromoacetate and chloroacetate. So, but for the activity, they have 5 times higher towards the fluoroacetate than the other 2. So this result was published in 2011 in a GACS journal. So, and then this study also proposed the catalytic residues and the catalytic mechanism of the fluoroacid dehalogenation. So we have the catalytic residue here as aspartate 110, histidine 280, and another aspartate 134. So and the catalytic mechanism starts with this undergoes SN2 mechanism, which means this aspartate will attack to the alpha carbon of the substrate and get replaced with fluorine ion. And the intermediate will be water hydrolyzed, assisted by the other catalytic residue, and then we will form this enzyme product complex. So the end product as glycolic acid. So, and also later in the 2017, this monthly Leeds lab in Germany, they also discovered this RP1163. They also accept the bulkier substrate, like the 2-fluoro-2-phenylacetate or the 2-fluoro-2-benzoic acetate. So they proposed the catalytic mechanism similar like RP1163, and the intermediate they formed this phenylacetate. And about this paper, they also find for the activity against the 2-fluoro-2-phenylacetate, they have like 5 to 10 times higher than the fluoroacetate. So which also indicated the fluoroacetate may not be the natural substrate for this enzyme. So, and out here in our collection, so we have these 8 enzymes. So they're bacterial defluorinases, they active on the fluoroacetate. So they belong to the family haloacid dehalogenases and fluoroacetate dehalogenases. So we were overexpressed in recombinant E. coli and purified for the activity screen. So here we have we selected 5 water-soluble fluorinated substrate: Fa, DFA, DFPA, and TFPA, and then PFOA. So these 5 water-soluble fluorinated substrate, we use our 8 enzyme to screen activity. So we all the enzymes, they have activity against fluoroacetate and defluoroacetate, especially for this DAR335. So they from the dichloromonas aromatic, and as well as for the NOS 0089 and POR 4478, they from the nostoc species and polymonas species. So these 3 were selected for the further study. And also back to the substrate. So we have this, you can see they have small activities on the DFPA and TFPA substrate, but there's no activity on the PFOA. So continue, we use the LCMS to analyze the final product. So for the fluoroacetate, we analyze the glycolic acid as an end product. For the difluoroacetate, we analyze glyoxylic acid as an end product. So, and we crystallize we plan to crystallize 3 structures of this candidate, but only 2 were successfully obtained, and by the molecular replacement. And these 2, the DAR335 and NOS 0089, they reveal the classic alpha beta hydrolase fold. So here, they have a codomain, and they centered with this beta sheet, and they have a alpha helices around, so on the both sides, and on the top they have a cap domain. So this cap domain is entirely covered by the small helices and connected by the flexible loop. So, and we also find this DAR335 have a bond of chloride ions right in the active center. So replace the fluoride ion. So they both enzymes, they form a dimer form, and they connected by the flexible loop. So this paper is published by our lab in 2023 in the FEBS journal. And also close look to the catalytic residue here. So because we already discovered the RP1163, so we have also here the catalytic residue, aspartate, histidine, and another aspartate here. So for the DAR335, we find this bond chloride ion, and which is closely interact with this key catalytic residue aspartate 107, and they also have this adjacent residue arginine 111. And we also looked at the fluoride stabilized residues, and we compared with the RP1163. So we have a tryptophan, histidine, and tyrosine here. So all this residue there give our understanding how this fluoride substrate interact with the inactive site. This is important, and we also crystallize the intermediate structure. So we took the one of the mutant. So the asparagine replaced the histidine at position 274. So this mutant, they somehow block the normal activity of the enzyme. So we are likely to capture this intermediate phase. So this aspartate and glycolate intermediate here. So this side on the right hand side, this electron density map confirm this intermediate. So and the surrounding residue, arginine 111 and arginine 108, they somehow stabilize this intermediate by interacting with the carbonyl group. And also we have checked this substrate access tunnel. So they're indicated by the red arrow. So here is you can see for this DAR335, we have a chloride ion right in the active center. And we also mapped this surface residue, and they are play an important role for the substrate recognition and accessing. So all these residues show they have a potential for the future engineering. So, and here we show the overall structure. So the overall picture of these three candidates and pink area show this substrate access tunnel. So from here you can see we have the tunnel start from this key catalytic residue aspartate and along the access to the surface. So all those three, they show the same thing. So, and we also propose the reaction mechanism for the defluoroacetate defluorination by the DAR335. So as we said, we have a proposed catalytic residue aspartate, histidine, and another aspartate. So when they react with the substrate difluoroacetate, they remove the first fluoride ion, undergoes the SN2 mechanism. So they form this intermediate to fluoroglycolate, and then the water helped to hydrolyze the intermediate and assisted by the catalytic residue here, and they form this fluorohydroxide acetate. And then the second defluorination reaction removed the second fluoride ion here, and to form this intermediate, and then intermediate dehydroxide acetate, and they can spontaneously dehydrated to the glyoxylic acid as the end product. So here we checked this structure. We did a structure analysis, and we proposed this catalytic mechanism for this defluorination. So we also measure this kinetics, enzyme kinetics. So they shows they have a relatively low substrate affinity, low substrate affinity. So which means we have a great potential to do the engineering to improve either the substrate affinity, or we can find the potential for other substrate degradation for these two enzymes. So we have this proposed protein engineering work. So this is planned collaboration work with Professor Weibong Shu, so in Germany. So because in our previous study, we use the set-directed mutagenesis, but we are not aiming to improve the activity. So we just want to find this intermediate structure and how this catalytic mechanism works. So now this time we're going to use the direct evolution, try to generate the mutant, randomly generate a mutant. And in the host lab, they have really good robotic platform and very well established methods. So, and we will use the method called split GFP technology. So our candidate select is DAR335 and RP1163. So we will fuse this GFP11 tag. And once when they complement with this GFP110, and they will give it a fluorescence, and only give the fluorescence when these two are together. So this method is provide the quantification of soluble protein content. The only soluble protein will give the fluorescence. And we have accurate identification of improved protein variants during the high throughput screening. So, and activity measurements can without the purification. So this we also, we combine this activity screen. We use, instead of using the pH sensitive, the phenol, so we use alizaline complex. So this is the complex that has the mental iron here. So it's a lanthanum in our case. So when the fluoride is here, we will replace the lanthanum and give the color change. So we currently accommodate these methods in the vivo state. So we already tested in the purified enzyme that works very well. And so we are accommodate and hopefully will be ready when we have this plan, before this planned collaboration work. So our lab is also welcome for the other collaboration on this PFAS study. And thanks for my supervisors and other team member, and big thank for the EBIC grant for the research support.